So good afternoon to all. So in the last class we have completed the process of gametogenesis. Okay, in that we have studied the spermatogenesis and oogenesis and the structure of the sperm. Okay, so next is the early development of the human. Early development of human. So in this topic we are going to study some of the aspects regarding the development of human. Okay, so synopsis in this, the following concepts or following aspects we are going to study, that is structure of an egg, then fertilization process, then cleavage, then morula, blastula and gastrula. So from structure of egg to gastrula, in your syllabus up to gastrula they have given. So we have to study all these processes. Okay, so first one, we'll study the structure of egg. So this is the diagram which showing the structure of ovum or the structure of human egg. Okay, so here see, you can see the different parts of the ovum here. First one is, that is the from top to bottom we are going to see the different parts of the ovum. So that is follicle cells or follicular cells. Next one is corona radiata. Then nucleus, central, that is a nucleolus. Another one is the dot like structure, that is the spacious structure inside the ovum, that is called as cytoplasm. And the layer, that is called as plasma membrane, inside inner layer. And the outermost layer, that is one more, there is a layer called as zona pellucida. And there is a space between inner and the outer layer, that is called as pre-vitelline space. So these are some different parts of the the layers. Means here especially there are membranes, we can call egg membranes anta kariತೀವಿ. There are 3 egg membranes. So outer one is corona radiata, and the outer one is corona radiata, and the middle one is zona pellucida, and the inner one is plasma membrane. So these are the 3 membranes of the ovum. Okay, the corona radiata is formed of what? Follicular cells. The follicular cells are present in the ovum. Those follicular cells are going to be form the outermost layer or the membrane, egg membrane, called as corona radiata. Then the zona pellucida corresponds to the vitelline membrane. So there is another membrane, that is the vitelline membrane, or it is also called as plasma membrane, inner membrane. So the space that is present between the zona pellucida and the plasma membrane or the vitelline membrane. So in between there is a space that is called as pre-vitelline space. Okay, the cytoplasm of the egg is called as, anybody, what is the name of the egg cytoplasm? The cytoplasm of the egg is called as ooplasm. What is called as ooplasm? Next, the ooplasm contains what? It contains, the ooplasm or the cytoplasm of the egg contains nucleus on one side of the egg, and the nucleus is usually in which form it is? Haploid or diploid. So it is in the form of haploid, because the male gamete is haploid and the female gamete is also haploid. So both combined together, or the union of male and a female gamete, that is called as zygote. Or union of male gamete and female gamete is called as the formation of the zygote is called as, the process is called as fertilization. So next we'll move to the next slide. In that there is explanation. So usually you can see here, an ovum is generally spherical shaped and non-motile gamete. It is not motile, cannot move automatically. So non-motile gamete with the yolky cytoplasm, means the cytoplasm contains yolk and it is enclosed in one or more egg envelopes. Already I have told there are 3 envelopes of the egg, isn't it? So it covers, or it is enclosed by a one layer or more layers or membranes. So size of the ovum varies in different animals. So in human beings also it is, the size is varies in different animals. It is species dependent, and depends upon the amount of yolk. So the size may be depend upon the amount of yolk. So size of the ovum, that is varies from how much, species to species, that is variation of the size taking place, isn't it? So it may be, it may be slightly larger than the spermatozoa. Okay, so we'll go to the next slide. So here human ovum is microlestal. Microlestal means it contains a very small amount of yolk, with large amount of cytoplasms. The cytoplasm is more, but the microlestal egg, there are different types of eggs based on the distribution of yolk. Microlestal, telolestal, mesolestal, megalestal, alestal, there are different types of eggs. So human egg or the ovum is the microlestal because it contains very less amount of yolk. So cytoplasm is differentiated into outer, smaller and transparent exoplasm or egg cortex, and the inner, larger and opaque endoplasm. Opaque colored endoplasm or ooplasm, we can call it as ooplasm. So egg cortex with some cytoskeletal structures are also present, that is microtubules and microfilaments. So according to Balansky in 1981, so these structures which are present in the egg. So another, there is a pigment granules and cortical granules are also present, and mucopolysaccharides are also present in the egg. Then endoplasm is with cell organelles. The endoplasm contains, endo means inside, the inner cytoplasm means the cytoplasm is differentiated outer ectoplasm and inner endoplasm, and endoplasm is with cell organelles. There are many cell organelles. Cell organelles are mitochondria, Golgi complex, like this. There are many cell organelles are there, no? So informosomes, that is tRNAs, histones, enzymes, so all these are present in the endoplasm. Okay, the nucleus of the ovum is large. So the nucleus, the size of the nucleus is large and blotted with nucleoplasm. There is a nucleoplasm also, and is called as germinal vesicle. What it is called? Germinal vesicle. And nucleus is eccentric. Eccentric means it is not exactly at the center. The nucleus is just away from the center. So it is eccentric in position. So the human ovum has a polarity. So polarity we can see in the human ovum. So the side of ovum with nucleus and the polar body is called as animal pole. Means the egg, complete egg is divided into two poles, that is animal pole and the vegetal pole. So the upper part, that is the called as animal pole, while the opposite side of the animal pole is called as vegetal pole. Vegetal we can call it because the yolk is present in the vegetal pole. Next, so already I discussed about the egg envelopes. The human ovum is surrounded by a number of egg envelopes, especially there are 3 we have discussed here: Whiteland membrane, It is a inner thin transparent membrane, and how it is formed means it is secreted by ovum itself. Okay. Zona pellucida is a second envelope. So it is a middle layer already I told, and it is a thick and transparent also as like a wetland membrane and non-cellular. It does not contains any cell. It is non-cellular ant karitara, and this has been secreted partly by follicular cells and other partly by oocyte. So half part of the uh, zona pellucida is by or formed by follicular cells, and another half part is formed by oocyte. Okay, so next last layer is the corona radiata. Already I have told it is the outermost membrane, and it is a thicker coat that is formed of radially elongated cells called as follicular cells. Okay, in between the vitelline membrane and the zona pellucida, there is another narrow space. So already I told that space is called as pre-vitelline space. Okay, so this is about the egg. So in the developmental biology means what we have to study? Developmental biology. Development we have to study. Biology means life. In the life of an human being or life of any organism, the steps which are involved in the developmental process. So from egg to an adult. Egg to an adult. ಯಾವುದೇ ಆರ್ಗನಿಸಮ್ ಇರಲಿ ಫ್ರಮ್ ಎಗ್ಲಿಂದ ಅಡಲ್ಟ್ ಆಗ್ಬೇಕು. ಅದಕ್ಕೆ ಏನೇನು ಪ್ರೊಸೆಸಸ್ ಬರ್ತಾವ ಅದಕ್ಕೆ ಡೆವಲಪ್ಮೆಂಟಲ್ ಬಯಾಲಜಿ ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ಅಬೌಟ್ ದಿ ಎಗ್, ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಎಗ್, ಫಸ್ಟ್ ಕಾನ್ಸೆಪ್ಟ್. ಸೊ ನೆಕ್ಸ್ಟ್ ಒನ್ ಈಸ್ ಫರ್ಟಿಲೈಜೇಷನ್. So what is fertilization? Fertilization is a process. It is also one of the process of development in which what happens? The zygote is formed. How the zygote is formed? So union of gametes. Union of gametes. Male gamete and the female gamete. Male gamete is spermatozoa and the female gamete is ovum. So both the union of both these gametes resulting in the formation of zygote, diploid zygote. Haploid, they become diploid zygote. So that is called as fertilization. Okay, so fertilization is sequence of events by which sperms fuses with the ovum from zygote occurs in oviduct ampulla. So fertilization is a sequence of events by which the sperm fuses with the ovum. That is what I have told already. Male gamete and female gamete, they combines together to form a diploid zygote. So that is called as fertilization. So where it is occurs, it occurs in the oviduct, where the fertilization takes place in the oviduct. Okay, oviduct naga fertilization ಆಗ್ತದೆ. ಫೆಲೋಪಿಯನ್ ಟ್ಯೂಬ್ ಅಂತಲೂ ಕರಿತೀವಿ ನಾವು ಅದಕ್ಕೆ. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಸೆಕೆಂಡ್ ಪಾಯಿಂಟ್ ಈಸ್ ಇಟ್ ಲಾಸ್ಟ್ ಅಬೌಟ್ 24 hours. ಸೊ ಎಷ್ಟು ಟೈಮ್ ಬೇಕು ಇದಕ್ಕೆ ಫರ್ಟಿಲೈಜೇಷನ್ಗೆ ಅಂದರೆ ಇಟ್ ಲಾಸ್ಟ್ ಅಬೌಟ್ 24 hours. Then third is spermatozoa pass into the tubular ampulla by movements of the sperm tail. So already we have discussed how the sperm is going to move. It is the movements of the sperm taken place by the help of the or with the help of the tail. From the tail region only the spermatozoa can move. So it passes into the tubular ampulla by the movements of the sperm or the sperm cell or sperm tail. So fourth point, sperms are viable for 12 to 48 hours after ejaculation. So sperm are viable means 12 to 48 hours they are viable after the ejaculation. So in the right side you are seeing the diagram, isn't it? In that diagram, so there are either outside you are seeing, so those are the sperms. Those sperms which are entering into the where they are entering into the oviduct, means they are entering and where the female gamete is present, and they will reach the female gamete and the fertilization takes place. So these are the number of sperms. Okay. So next one is the ovum passes into the tubular ampulla by beating action of the cilia and muscular contraction. So it is from the ovary only the ovum is released. So that from the ovary it has to come to the uterus or the tubular ampulla, that is fallopian tube oviduct. So how it comes from ovary to the fallopian tube? By the beating action of the cilia. Cilia, it is a hair-like structure that is helpful for the movement. Cilia and muscular contraction. Okay, so the egg is viable for 12 to 24 hours after ejaculation. Then spermatozoa are reduced in number during the passage. Means the through that already in that diagram you have seen, through the passage the number of spermatozoa are reduced in number during the passage through the female reproductive tract. So there are many sperms. So in those sperms the number of spermatozoa is going to be reduced in number. So at the time of the passage through the female reproductive tract. So why the number is reduced? Due to the elimination of abnormal and poorly motile sperms. So sperms during the ejaculation from the man there are number of spermatozoa are released, but all those spermatozoa cannot reach the ovum because some of the sperms which are non-motile and they are abnormal also and they are poorly developed or poorly abnormal condition or in the non-motile state. Hence all the sperms are not reached and they lost during the passage in the female reproductive tract. So capacitation is the enzymatic removal of the glycocalyx coat from the sperm membrane over the acrosome. Here the capacitation process takes place. So it is one of the enzymatic removal of the glycocalyx coat from the sperm from membrane over the acrosome. Then it takes about 7 hours to capacitate. So how much the capacitation process needs? It takes 7 hours. And the sperm cells must make their way to the uterine tube for fertilization to be possible. So here the sperm cell must enter. They must enter and the uterine tube for the fertilization. Then only the fertilization occurs. Otherwise there is no fertilization takes place without the male gamete. The sperm and the egg join to form the zygote. Hence the fertilization takes place, the first cell of a new individual. Okay. Next, the human zygote is synkaryon because it contains two pronuclei, because one already present in the ovum and another nuclei comes from the sperm, that is a male gamete. So it contains, the zygote contains two pronuclei because it is a diploid in nature, and zygote results of fusion of DNA from the sperm and eggs. So fertilization occurs in the fallopian tube, already I told, in the fallopian tube, and zygote begins rapid mitotic cell division. So here what happens, the zygote undergoes the mitotic divisions rapidly, and the zygote is beginning of human development. So when the zygote fertilization occurs, the process of development starts. The zygote floats down from the fallopian tube towards the uterus. So here from the zygote formation the cleavage will be produced. So this is about the fertilization. Next we are going to study, that is cleavage. So cleavage means what? Cleavage means rapid repeated mitotic division of the zygote. Means rapid repeated mitotic division of the zygote. Already zygote is there. So that zygote undergoes repeated mitotic divisions or successive rapid mitotic divisions without growth of daughter cells called as blastomeres. Means the zygote undergoes division rapidly, repeatedly, and they forms the blastomeres. and these blastomeres retain totipotentiality. Totipotency ಅಂತ ಕರಿತೀವಿ. ಅಂದರೆ ಇಟ್ ಈಸ್ ದ ಕೆಪಾಸಿಟಿ ಆಫ್ ದ ಸೆಲ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ಟೋಟಿಪೊಟೆನ್ಸಿ ಅಂತಾನೆ, ಅಂದರೆ ಎನಿ ಟೈಪ್ ಆಫ್ ದ ಸೆಲ್. Next, it occurs during its passage through the fallopian tube, that is the oviduct, to the uterus, and it is a holoblastic. Holoblastic cleavage ಅಂತ ಕರಿತೀವಿ, where the complete egg divides. That is into two blastomeres. ಅದಕ್ಕೆ ಏನಂತಾರೆ holoblastic ಅಂತ ಕರೀತಾರೆ. holoblastic means the complete egg divide ಆಗ್ತದೆ. ಇಲ್ಲಿ ಏನು ಬರ್ತಾವೆ, ಇದರ ನಂಬರ್ ಆಫ್ ಕ್ಲೀವೇಜಸ್ ಬರ್ತಾವೆ, ಅಂದ್ರೆ ಬ್ಲಾಸ್ಟೋಮಿಯರ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ದ ಫಸ್ಟ್ ಕ್ಲೀವೇಜ್ ಅಕರ್ಸ್ ಅಬೌಟ್ 30 hours ಆಫ್ಟರ್ ಫರ್ಟಿಲೈಸೇಷನ್. ಫಸ್ಟ್ ಕ್ಲೀವೇಜ್ ಯಾವಾಗ ಆಗ್ತದೆ? ಸೊ ಇನ್ ದಿ ಲಾಸ್ಟ್ ಸ್ಲೈಡ್ ಯು ಸೀನ್ ದಿ 2 celled stage, 4 celled stage, 8 celled stage, 16 celled stage, and marula. And like that again, after marula there is a blastula. Like that there is a development takes place. So here a cleavage 2 cell, 4 cell, 8 cell, 16 cell. ಇಲ್ಲಿ ತನ ಅರ್ಲಿ ಕ್ಲೀವೇಜ್ ಈಸ್ ಅಪ್ ಟು 8 cell ಆರ್ 16 cell ತನ ಆಗ್ತದೆ. ಆಮೇಲೆ 16 cell ನೆಕ್ಸ್ಟ್ ಈಸ್ ಲೇಟ್ ಕ್ಲೀವೇಜ್ ಸ್ಟಾರ್ಟ್ ಆಗ್ತದೆ. ಸೊ ದಿಸ್ ಇಸ್ ದ ಡೈಗ್ರಾಮ್. ಸೊ ದ ಫಸ್ಟ್ ಕ್ಲೀವೇಜ್ ಅಕರ್ಸ್ ಅಬೌಟ್ 30 hours ಆಫ್ಟರ್ ದಿ ಫರ್ಟಿಲೈಸೇಷನ್. ವೆನ್ ಇಟ್ ಅಕರ್ಸ್ ಆಫ್ಟರ್ 30 hours, ಫರ್ಟಿಲೈಸೇಷನ್ ಆದಮೇಲೆ 30 hours ಆದಮೇಲೆ ಫಸ್ಟ್ ಕ್ಲೀವೇಜ್ ಆಗ್ತದೆ. ದೆನ್ ದ ಸೆಕೆಂಡ್ ಕ್ಲೀವೇಜ್ ಅಕರ್ಸ್ ಅಬೌಟ್ 60 hours. 60 hours ಆದಮೇಲೆ ಸೆಕೆಂಡ್ ಕ್ಲೀವೇಜ್ ಆಗ್ತದೆ ಆಫ್ಟರ್ ಫರ್ಟಿಲೈಸೇಷನ್. And the third cleavage occurs after 72 hours of the fertilization. ನೋಡಿ ಯಾವಾಗ ಆಗ್ತದೆ ಅಂತ ನೆನ್ಪಿಟ್ಕೋಬೇಕು. ದಟ್ ಈಸ್ ಅಂದ್ರೆ ಥರ್ಡ್ ಕ್ಲೀವೇಜ್ ಆದಮೇಲೆ ಇಲ್ಲಿ ಥರ್ಡ್ ಕ್ಲೀವೇಜ್ ಆಗ್ತದೆ. ಫಸ್ಟ್ 2, ಆಮೇಲೆ 2 ಹೋಗಿ ನೆಕ್ಸ್ಟ್ 4 ಹೋಗಿ ನೆಕ್ಸ್ಟ್ 8. ಸೊ ಇದು 72 ತನಕ ಆಗ್ತದೆ. ಮುಂದೆ ಏನಾಗ್ತಪ್ಪ ಅಂದ್ರೆ 16 ಸೆಲ್ಲು, ಆಮೇಲೆ ನಂಬರ್ ಆಫ್ ಸೆಲ್ಸ್ ಆಗೋತ ಹೋಗ್ತಾವೆ, ಅಲ್ಲಿ ಚೇಂಜ್ ಆಗೋತ ಹೋಗ್ತಾವೆ. ಆಮೇಲೆ 16 hours 16 cells ಆದಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಮಾರುಲಾ ಬರ್ತದೆ. ದಟ್ ಈಸ್ 32 cells ಬರ್ತದೆ. ಅವಾಗ ಮಾರುಲಾ ಅಂತ ಕರಿತೀವಿ ಅದಕ್ಕೆ, 32 cell ಸ್ಟೇಜಿಗೆ. ಸೊ ದಿಸ್ ಈಸ್ ಅ ಮಾರುಲಾ. ಸೊ ಸಾಲಿಡ್ ಮಾರುಲಾ ಅಂದರೆ ಸಾಲಿಡ್, ಇಟ್ ಈಸ್ ಎ ಸಾಲಿಡ್ ಸ್ಟ್ರಕ್ಚರ್. ಇಟ್ ಈಸ್ ಮಾರುಲಾ ಅಂದರೆ 32 celled stage solid marula that develops into the hollow fluid filled blastula. Next, after the marula there is another one stage that is called as the next stage is blastula. So the embryo will develop from the inner cell mass or embryonic disc. It occurs about 96 hours after fertilization. So when it occurs, when it is going to be formed means after 96 hours after fertilization. So and it consists of 8 to 16 blastomeres. So in the marula you are going to see 8 to 16 blastomeres. In the last diagram you can see late cleavage. Next, marula. So after marula there is a blastula, means formation of blastula from the marula is called as blastulation ಅಂತ ಕರೀತಾರೆ. So after the marula there is a blastula. So it occurs 5 days after fertilization. ಅಂದರೆ 5 ದಿವಸಗಳಾದಮೇಲೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಆದಮೇಲೆ this structure is going to be formed. So it is produced by repeated mitotic division of zygote. ಹೆಂಗೆ ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ ಇದು ಅಂದರೆ ಇಟ್ ಈಸ್ ಫಾರ್ಮ್ಡ್ ಬೈ ದಿ ರಿಪೀಟೆಡ್ಲಿ mitotic division of the zygote. A ball of more than 64 cells surrounding a fluid cavity called as blastocele, means 64 celled 64 cell ಸ್ಟೇಜ್ ಇದ್ದಾಗ ಅದಕ್ಕೆ ನಾವೇನು ಕರಿತೀವಿ ಬ್ಲಾಸ್ಟುಲಾ ಅಂತ ಕರಿತೀವಿ. ಆ್ಯಂಡ್ ಇನ್ ದಿಸ್ ಬ್ಲಾಸ್ಟುಲಾ ದೆರ್ ಇಸ್ ಎ ಕ್ಯಾವಿಟಿ or the cavity or the space. So that cavity in the blastula is called as blastocele. The 2 marks question may appears. What is blastocele? Then you have to write it is the cavity present in the blastula is called as blastocele. In the diagram also you are going to see in the right side. Okay, blastocele, that is a cavity inside the blastula. So this cavity is filled with the fluid. One of the fluid, that is fluid filled cavity we can call. The human blastula is called as blastocyst. What is the name of the blastula in the human being is called as blastocyst. The blastocyst the ಏನಂದ್ರೆ ಬ್ಲಾಸ್ಟೋಸಿಸ್ಟ್ ಅಂದರೆ ಏನು ಅಂದರೆ ದ fastening of embryo to wall of the uterus is called as implantation. Means what is implantation? In the next topic it is there in your syllabus also. Implantation ಅಂದರೆ attachment to the attachment of the blastocyst to the uterine wall. So that is a implantation. So implantation occurs 7 days after fertilization. So after 7 days the fertilization occurs, means the attachment of the blastocyst to the uterine wall takes place. So this process is called as implantation, and it occurs, or the implantation occurs after 7 days of the fertilization. And where it is taken place? It is taking place in the uterus. Okay, so a diagram you can see, I am zooming it, you can see that there is a layer of cells, these are called as blastomeres, and there is a middle cavity called as blastocoel, and there is a inner cell mass and the trophoblast. So that is the outermost layer. So this is about the complete blastula. So then last one is the gastrula. So in the right side also you are seeing the diagram, that is gastrula. Here is the gastrula, there is a 3 germ layers are going to be formed. In the blastula there is only one layer, that is called as blastoderm, but in the gastrula there are 3 layers: ectoderm, endoderm and mesoderm. So 3 germ layers are going to be seen in the gastrula. From these germ layers only the different organs are going to be formed. Organogenesis antino, the process of formation of organs is called as organogenesis. So these 3 layers are going to form the different organs in our body. Okay, so blastula to gastrula, blastulalinda gastrula ಆಗ್ತದೆ, that process is called as gastrulation ಅಂತ ಕರಿತೀವಿ. So you are going to see here in the diagram also ectoderm, endoderm and archenteron, means the cavity present in the gastrula is called as archenteron, otherwise it is also called as gastrula ಅಂತನೂ ನಾವು ಕರಿತೀವಿ. Okay, so here is a different, in the left side there is a diagram, both the diagrams are shown, that is blastula also. Here the blastula is gastrula, both are shown here. So you just differentiate between gastrula and blastula. In the blastula there is a cavity called blastocoel, and in the gastrula there is a cavity called as gastrocoel, or called as archenteron, isn't it? And there is a ectoderm, endoderm and mesoderm. There are 3 layers. Okay, one by one the points we have to discuss. So it involves, means the gastrula involves a series of cell migrations to positions where they will form the primary cell layers. Here the movements of the cells, means which cells? Blastomeres are migrated. Migration of the cells taking place here. So that is called as morphogenetic movements. The movements of the cells or blastomere, that is called as morphogenetic movements. So these going to be form the primary cell layers or the germ layers. So that is ectoderm, endoderm and mesoderm. So the ectoderm is the first outermost layer that forms the outer layer, and it forms there means from the ectoderm, already I have told the organogenesis process. Organogenesis means ಅಂದ್ರೆ ಏನು, ಡಿಫ್ರೆಂಟ್ ಆರ್ಗನ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗೋದು ಬಾಡಿ ಒಳಗೆ. ಸೊ ಆರ್ಗನ್ಸ್ ಎಲ್ಲ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕಾಯ್ತು ಅಂತಂದ್ರೆ ವಿ ನೀಡ್ 3 germ layers. So from these 3 germ layers only different organs are going to be formed. Okay, so first ectoderm ಏನದಪ್ಪ ಅಂದ್ರೆ ಔಟರ್ ಮೋಸ್ಟ್ ಲೇಯರ್ ಅದ, ಆ್ಯಂಡ್ ಇದು ಏನು ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತದ? ಇಟ್ ಫಾರ್ಮ್ಸ್ ಸ್ಕಿನ್, hairs are going to be formed, skin is going to be formed, sweat glands ಅಂದರೆ ಏನಾಗ್ತಾವೆ, ಈಗ ನೀರು ಬಿಡ್ತಾ ಇರ್ತಾವಲ್ಲ, ನಮಗೆ ಸ್ವೆಟ್ಟಿಂಗ್ ಆಗ್ತಾ ಇಲ್ಲ, ಅದಕ್ಕೇನಂತ ಕರೀತಾರೆ ಸ್ವೆಟ್ ಗ್ಲ್ಯಾಂಡ್ಸ್, ಆ್ಯಂಡ್ ಬ್ರೈನ್, ನರ್ವಸ್ ಸಿಸ್ಟಮ್, epithelium. So all these structures are going to be formed from the ectoderm. Okay, just you have to remember the nervous system, skin, hairs, sweat glands, all these are formed from the ectoderm. It means endoderm. Endo means inside. So inner layer means it forms the inner layer and it forms the digestive system. So in the body, digestive system is going to be formed from the endoderm, and respiratory system, liver, pancreas, bladder, and endocrine glands. Such as endocrine glands are ductless glands. There is another glands in our body. What are those? What? Exocrine glands. Exocrine glands are duct glands. They do not secrete any hormones. They secrete only some juice, like salivary glands. Example, they are the exocrine glands. So endocrine glands are the glands which secretes the hormones in the bloodstream. So they are thyroid gland, parathyroid glands. There are other glands of endocrine: pituitary gland, adrenal gland, pineal gland. These are all endocrine glands because they secrete the hormones. So all these all these glands and respiratory system, digestive system, all these are going to be formed by the inner layer of endoderm. Then mesoderm, it is in between two layers, ectoderm and endoderm. Endoderm, there is another layer, that is a mesoderm. So third germ layer. So it forms the middle layer and it forms body muscles. So muscles are going to be formed and cartilage is going to be formed, bones are going to be formed, blood is going to form, and reproductive system. That is, reproductive system means in the male testes, in the female ovary, like this. These are going to be from reproductive structures, organs, and kidneys also formed from the mesoderm. So all these three germ layers going to form the different organs or different structures in our body. Okay, already we have studied in the first year also, that is levels of organization. ಏನೇನು ಫಾರ್ಮೇಷನ್ ಆಗ್ತಾವ, ಸೆಲ್ combines together to form tissue is going to form organ combines to form the organ systems, like organ systems combine together to form the organism. ಸೊ ಹಂಗೆ ಆಗ್ತಾ ಇರ್ತದೆ. ಸೊ ಇದು ಆರ್ಗನ್ಸ್ ಎಲ್ಲ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತದೆ. ಆರ್ಗನ್ಸ್ಗಳು ಮುಂದೆ ಆರ್ಗನ್ ಸಿಸ್ಟಮ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತಾವೆ, ಆರ್ಗನ್ ಸಿಸ್ಟಮು ಮುಂದೆ ಆರ್ಗನಿಸಮ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತದೆ. ಓಕೆ, ದಿಸ್ ಎಂಡ್ಸ್ ಅಬೌಟ್ ದಿ early development of the human. So what we have studied, first we have studied the structure of egg, because the development starts from the egg, and after the egg there is a fertilization, union of gametes. After the fertilization there is a cleavage, a rapid repeated mitotic division of the zygote called cleavage. 2 cell, 4 cell, 8 cell, 16 cell, 32 cell. Then comes the morula, and morula we have studied solid cell, and next is called as blastula. There is a blastocele is present, blastocele jelly, blastula fluid, like fluid is fluid within the blastocele. So blastula you have to study. After blastula there is another stage called as gastrula. Don't confuse with the gastrula and blastula, and blastocele and gastrocele. So blastocele, where the blastocele is present? Blastocele is present in the blastula. Then gastrocele is present in the gastrula. Okay, gastrula is also called as what? Archenteron. Okay, right. So where the three germ layers are present? Blastula or gastrula? Gastrula. Okay, good. In the gastrula there are three germ layers. What are those layers? Ectoderm, endoderm, and mesoderm. So all these are three layers of the gastrula. So what is organogenesis? The process of formation of different organs. So that is called as organogenesis. So from egg to adult, that is a development stages or developmental biology, you can call. So this is about the human development. So if you have any questions you can ask me, otherwise we'll finish up this. You have any doubts? Don't have any doubts? Okay, so the next class we are going to study the frog development. So human development you have studied, next the frog development. So that is, there is also same early development from egg to gastrula blastula ಎಲ್ಲ ಸ್ಟಡಿ ಮಾಡಬೇಕು. ಆ್ಯಂಡ್ ಆಲ್ಸೋ ದೇರ್ ಈಸ್ ಸಮ್ ಅನದರ್ ಎಕ್ಸ್ಟ್ರಾ ಕಾನ್ಸೆಪ್ಟ್ ದಟ್ ಈಸ್ ಅ ನ್ಯೂ ನೋಟೋಜೆನೆಸಿಸ್, ನೋಟೋಕಾಡ್ ಫಾರ್ಮೇಷನ್ ಆಗೋದೆಲ್ಲ ಓದಬೇಕು. ಸೊ ದಟ್ ವಿಲ್ ಸಿ ಇನ್ ದಿ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್. ಓಕೆ 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 you write some assignments on this topic. If you want, I will send the notes, and you write structure of blastula, draw with the help of diagram, explain the structure of egg, human egg, and gastrula, the process of gastrula, explain the process of gastrula. So you have to write some assignments also. Okay. Thank you.